From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 29th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 56th episode of Matters Microbial. Thanks so much for listening and/or watching and for spreading the good microbial word. I need all the PR I can get. As I record this podcast, classes begin at my institution next week. I have a new crop of budding micronauts to proselytize into microbial centrism, and I look forward to it. First, I well remember many years ago teaching my first lecture in microbiology to a group of students. At one point, I said that we were all emitting a cloud of microbes as we breathed or spoke. This horrified one student who rushed home, painted what you see now, and sent it to me. Because we do indeed exist within a microbial cloud after all. I have long been known to hand out various items to my new micronauts to encourage a microbial perspective, such as customized M&Ms seen here. Notice the four mottos: Quality Quorum, Microbes Rule, First Evolved, and Last Extinct. Also in the first week, I have students select a microbial pal to write a page about as a first assignment, and I use a sticker, and you can actually see it on the screen here. I have done my best to select strange and unusual and fascinating microbes. Artwork is by the sublime Ethan Kokic. To see to it that each student gets their own microbial pal, I hand out microbial keychains for them to select that Dr. Jennifer Quinn and I designed and 3D printed. Naturally, they glow in the dark. I cannot wait to share with you what my students uncover this semester. It is always a pleasure. One of my mottos from microbiology is First Evolved, Last Extinct. But what do we know about early life on Earth? After all, it was a very different environment, and the ocean was far different from today. Also, I personally have a long-standing interest in marine microbiology, and not just about luminous microorganisms, by the way. So it's a real pleasure to introduce Dr. Carolina Martinez Gutierrez of the Department of Earth Science at the University of California, Santa Barbara. She'll tell us about the story of her science, famous microbes you may not have heard of, and how to study microbial life in the early ocean. Carolina, welcome to the Quality Quorum for today. It's great to see you.
Hi, Mark. It's a pleasure to be here. Thank you so much for the invitation.
Isn't it fun?
Thank you so much.
Isn't it fun how we met at ASM and here you are?
Yeah, it was a couple months ago.
I know.
Yeah, it's amazing.
So I think it's really great to hear from you, and you're starting your new position. How exciting is it?
It is amazing. I cannot wait to have students here, but unfortunately, they will only come until a few months from now. My postdoc is coming soon, so we will start the research going very soon, which makes me super, super excited. Yeah.
It's really exciting on the one hand, but it's daunting because of all the things you have to do to get a lab up and running.
Yes.
I mean, what do you order? And I don't know. It used to be at the University of California, you had to bid everything several times before you ordered anything. I hope they've made it easier.
I hope so too. Luckily, for the work that we will start doing, we only need good computers, and those are very easy to order. So, well, relatively easy. So, yeah, I cannot wait to start things going.
Oh, it means so much to see microbial enthusiasm. So let's go ahead and get started. And I think a good place to start, because you and I communicate a little bit about what you'd like to cover today. Would you care to talk about how the ocean, we talk a lot about microbiomes in this podcast, yes, but the ocean has one too. And I want people to remember microbiome isn't just in association with another organism, which is how many people use it, but it can return to environments. So would you like to discuss a little bit about how the microbiome of the ocean interacts with the geochemistry of our planet?
Yeah, I love that topic. And a few years ago, I read that the ocean microbiome is the largest microbiome on Earth. And you can imagine that's kind of obvious because the ocean is so huge. It covers 70% of Earth's surface. So, of course, the microbes that are living there have very important implications for Earth's geochemistry. So, for example, some microbes are involved in carbon fixation, which is very important for the productivity of the ocean and ultimately land. So, land, terrestrial environments really depend on what's going on in the ocean. Some oceans are implicated in the recycling of this organic matter. Some organisms are producing oxygen because photosynthesis is also happening in the ocean. And a lot of the oxygen we breathe in is coming from the ocean. No matter where you are on planet Earth, you will have a relationship with the ocean, which is fascinating because I love the ocean. And even if I'm far from it, I know that I still have some interaction with it. Um, what other things? They recycle sulfur, nitrogen. So, for example, there are some organisms that can fix nitrogen. Microorganisms are the only ones that have the capacity to fix nitrogen. Um, so, there are many microorganisms living, for example, in the open ocean that are very far from land environments that have the role of fixing nitrogen and keeping these communities going on. So yeah, microorganisms are extremely important in the ocean, and really we all depend on them and the activities they are having in the ocean. Yeah.
One of the challenges that I face when I teach about this is there's been, and I mean no offense to anyone when I say this, a certain degree of eukaryocentrism that's been like pushed in our educational systems, so that people will tell me, well, oxygen is made by plants, and that's not really true.
Yeah, that's absolutely correct. And of course, plants have a very important role in the
Hmm.
production of oxygen, but microorganisms living in the ocean have a very, very important role as well. Um, so yeah, that's why we should care about these microbes as well, not only the big things, right?
Absolutely. And I even go further than that when I point out that chloroplasts are clearly descended from cyanobacteria. So,
Exactly.
and this gets me the whole microbial centrism thing. People sometimes call me names about it, but I'm at an age where I don't care because I do think it's at the center of our biosphere.
Yes, exactly. Even chloroplasts living inside the eukaryotic cells of plants or algae. Well, algae are sometimes considered plants as well,
I
depending
know.
on who you ask. Uh,
And it's so funny because we tend to stick with just a couple of model systems everywhere in science, and the diversity of the microbial world lends itself to us finding out interesting things and thinking about things in other ways that we're not used to. And that's one of the things that I kind of really push hard on my students, because I only have one course in microbiology, and you and I both know it requires its own major.
yes, yeah, I do agree. Yes.
So you were going to also mention a few specific members of the, I'm going to call it the oceanic microbiome that people may not have
Mhm.
heard of. And I included both of those in the organisms that students can potentially study in my class next week. You'll be glad to know.
Oh, great. Yeah, as I mentioned, the ocean is extremely huge. It covers a huge surface of our planet. And there are some areas in the ocean that are particularly large. And these are the oceanic gyres that are in the middle of the ocean. For example, we have two in the Pacific Ocean, two in the Atlantic Ocean, and there are others in other parts of our planet, but they are extremely large and they are very important because they are very far from land, meaning that there is a very small input of nutrients coming from terrestrial environments, and they pretty much depend on what's going on there. So there are a few microorganisms that are extremely abundant in these type of environments, and some people are very familiar with these names. So, for example, Prochlorococcus is one of them. Everyone loves Prochlorococcus. And this is a very interesting microorganism because they have a little bit of strange pigments. So, they are relatively newly discovered because the techniques that people were using to discover photosynthetic microbes were not working on this particular organism. The other one is SAR11, or pelagibacteria or pelagibacteriales, depending on the classification you are using. They are very abundant in the ocean as well. And they are not phototrophs, so they don't have any pigments. They are heterotrophs, meaning that they consume the organic matter that other organisms are producing. And Prochlorococcus and SAR11 are very abundant in the ocean. Some people suggest that they are the most abundant organisms on Earth, which is crazy to me. They represent a lot of biomass in marine ecosystems. And they are very different because although it's a little bit of an strange case because they are located in completely different areas of the tree of life. One of them is a cyanobacterium in the case of Prochlorococcus, and the other one is a protobacterial microbe. So this is another group of microbes, but they have converged in certain traits. For example, they have very small genomes. Their genomes are extremely small compared with, for example, E. coli or Salmonella, a microbe that very well studied. They have very low GC content. This is guanine cytosine in their genomes. So they have these very strange genomic traits that are probably related with the environmental conditions in which they live. But yeah, these organisms are extremely interesting, extremely important for the functioning of the ocean. And there are other organisms that are, for example, located in archaea. One of my favorite ones is ammonia oxidizing archaea. They are also very abundant in the ocean, and they have very important roles. For example, they transform ammonium into nitrate. Nitrate is very, very important for the functioning of the ocean, not only for other microorganisms living around them, but also for eukaryotes. So everyone should love ammonia oxidizing archaea for that reason.
No, I agree completely. I love these three well, the last one's more of a group, but the three organisms or groupings you've talked about are really important. And there's a term that I've heard applied to SAR11, the pelagibacter, that they've been genomically streamlined. And
Yes.
and they have, but I would call it Darwinowing because the hand of Darwin is on all that lives, and we tend to really put our own opinions onto environments, and the microbes don't care. So living in these
Exactly.
very oligotrophic environments, very low in nutrients, they've made these very fundamental adaptations, and they've simplified down to the bare bones of what they've needed. And as a result, as you know, and hopefully I'll put a link in the show notes to this, the pelagibacter are actually quite difficult to grow because they can't handle high levels of nutrients at all.
Yeah, and that has to do with their small genomes.
Yes.
So one of the main features of the open ocean is that overall is a very stable environment. So there are no dramatic changes in terms of nutrients concentrations, except when there are, for example, upwellings in some regions of the ocean. But that allows microbes to be extremely adapted or have a very specific type of metabolism that is specific for that type of environment. That's one of the reasons why they don't have many genes in their genomes, because you don't need them. There are not many changes. Why would you have this extra fancy machinery in your genome if you don't really need it? Like, it's kind of a burden to have all these genes that you don't really need. So selection has got rid of so many genes that are not needed in this environment. And when we try to bring these microbes into the lab, of course they will not like the conditions that we have in the lab, because they have been evolving for millions of years in other conditions. So yeah, that's the reason why SAR11 is very hard to grow in under lab conditions.
It's very interesting, and I tell students most of what we know about microbes pretty much predicated on the study of Escherichia E. coli. And the problem is E. coli's natural habitat was the lower gut, and most of the E. coli that we use in the laboratory was in fact derived from one sample, Stanford University, 1912. So the
Oh,
point
wow.
is, the point is there are others, but it's interesting because K12 is the one that does gene transfer, and some of the other ones, type B, doesn't. So
Mhm.
think about if they'd tried to do all of E. coli genetics not with K12, but this other
Yeah,
strain, they would have had a much more difficult time. But the reason I bring this up is
exactly.
that it's a lot of our ideas are based on what works for E. coli, which is used to a copiotrophic, lots of nutrients environment. And it really colors our thoughts. And I call this coli centricity because actually these organisms are really good at adapting themselves to the ecophysiology of their environment. And it's really fun to think about because, well, okay, it's fun for me. Students trying to study rules don't like it, but I love how variable things are. I always have.
Yes, yes. Well, the opposite happens in the ocean. Very stable environment, not many things. Well, this is of course relative, right, to what,
Mhm.
for example, is happening in the gut. Um, Very stable environment, not many things going on. One of the things that I could think that could disturb this is viruses, for example. They are everywhere all the time attacking these microbes. And that's one of the questions, like how can these microbes deal with the viruses that are abundant in the ocean, right? That's an ongoing field of research, of course.
And it just fascinates me. I know Pelagibacter has viruses.
Yes,
But honestly, I suspect every living thing that's a cellular
exactly.
organism has viruses. Let's be clear.
Yeah.
And I also want to just take a moment. You gave me a wonderful link to the discovery of Prochlorococcus by a heroine in that particular area. And I so look forward and I
Yes.
urge people to watch that video because Peggy Chisholm is
Yeah.
awesome.
Yeah. She's amazing. Um, yeah, I'm honestly in love with these microbes. I love them. I love to study their evolution and how they became how they are.
Yeah.
That's a question that fascinates me.
Carolina, I've been reading some really disturbing articles in the local media about a possible change in some of the currents in the North Atlantic, leading to a collapse of a particular current, and the thought that that might cause really serious climatic change in Europe. And surely that's going to cause all kinds of change to local ecosystems. Is there any evidence on how this might, or any thoughts you might have on how this might change kind of the microbiome of the oceans, for lack of a better term?
Yeah, that's a very good question. And I will say that the main impact could be in the way how nutrients are recycled. Because, for example, if temperature increases dramatically, there will be a stronger stratification of the ocean, and that will prevent nutrients that are recycled in the deeper ocean from going up to be used again by microbes. And ultimately to be used by larger forms of life in the ocean. So yeah, that will be one of the main effects of such a dramatic change.
So that brings us a little bit to what the topic is today, which is the idea of echoes of the ancient past. And if you think about it, a good place to start is to talk about the geological history of the ocean. Most of us at some time or the other have seen the oh, I shouldn't say boring, but they were the boring videos of how like the different periods of life on Earth, and we're sitting there in the fourth grade like this. And you wouldn't think it would be much more interesting in the ocean, but you'd be wrong.
Yes. So yeah, you're right. The ocean since its origins, which is at least 4 billion years. The ocean is at least 4 billion years. You can imagine there have been so many changes. And life, I think the oldest evidence of life is 3.4 billion years old. These are the oldest fossils we have discovered. And but you can imagine that's a minimum for the age of life. So life could be way older than that. And some evidence based on molecular dating and the analysis of genomic data have shown that life could be maybe 4 billion years old. So life is extremely ancient. The ocean is extremely ancient, and both of them have been going through dramatic changes since their origins. So one of my favorite dramatic changes is the Great Oxidation Event. And this is when oxygen became available in the atmosphere in high concentrations for the first time. This we think was a result of the metabolic activities of cyanobacteria, which were the first microbes able to do photosynthesis that was based on water as electron donors, because before we had photosynthesis, but that type of photosynthesis was not producing oxygen. And this new revolutionized type of photosynthesis could produce oxygen, which was very bad news for microbes back in the day, because oxygen is very reactive. It reacts with everything, it oxidizes everything. And although there is no evidence of that, we think that there was a huge mass extinction event of many microbes that haven't seen oxygen in high concentrations in the past, and their metabolism was adapted to work without oxygen. And the surface of the ocean became partially oxygenated. Um, and after this large event, the Great Oxidation Event, there is not really an event, because we think it was more like a period in which oxygen started accumulating in the atmosphere and in the surface of the ocean. After that period of oxygenation, there was an ups and downs in terms of oxygen concentrations. So again, very dramatic changes happening in the ocean. And interestingly, the lower part of the ocean, or the deeper part of the ocean, was oxygen free for a very long period of Earth's history. Only about 500 million years ago is when the deep ocean became oxygenated. And that's very interesting, because that's a very short, I mean, like, if you don't study these time scales, that might look like a lot of time, but in terms of the history of Earth, that's not much. So yeah, that's one interesting thing that happened in the ocean around 2.4 billion years ago. Also, during that time, there was a snowball Earth event. That means that even regions near the equator became frozen. And again, big changes for the ocean. I bet the microbes in the ocean were not having fun when this happened. So there were so many things happening. Around 700 million years ago, there were multiple snowball Earth events. The same happened, and this had to do with changes in the productivity of the ocean. When productivity increases, CO2 decreases, and that causes a lower temperature on Earth. So yeah, it's very interesting to see, or to think about, how the ocean has been going through these dramatic changes throughout its history. And something that really fascinates me is how microbes responded to those changes, because some of these microbes that are very important for the ocean today are extremely ancient. So for example, SAR-202, which is one of my favorite microbes as well. Well, all of them are my favorite ones. SAR-202 is included in the chloroflexi group. They are extremely ancient. They are like at least 2.4 billion years old, and they are still around us. And they are very important for the recycling of sulfur, for example, for sulfur metabolism. How did they survive? Like, when these things were happening, like, that's something that really fascinates me. How did all these ancient microbes managed to go through these changes? What were the adaptations they acquired? And finally, one of the main changes, at least one of my favorite ones, was the emergence of eukaryotic marine microbes and marine microscopic life. And that happened, well, we have strong evidence that they happen around the end of the Proterozoic Aeon, which is around 0.5 billion years old. They were there for sure around that time, and but they were there before, but maybe not in very high abundance, around 0.7 Yeah, they were there 0.7, 0.5 during that period. They became very abundant. Um, but that changed the dynamics of that geochemistry of the ocean, because instead of you being the only one producing oxygen or fixing carbon or fixing nitrogen, there were other guys fixing carbon as well. So the dynamics of the ocean changed dramatically because of the presence of eukaryotes.
So this is why you've been saying that the Precambrian era is the most interesting to you.
Yes, exactly.
And I want to say that, you know, most people, if they had a time machine and they were going backward through time, and we've all seen the movies about that, they keep forgetting that the majority of time life was on Earth, it was microbial.
Exactly,
And
exactly.
and I tell my students, imagine if there was some microbe that started appearing that created this terribly toxic gas,
Mhm.
and it was growing faster than everything else, because that's
Yes,
what happened.
exactly.
and
Yeah,
ox and
that's exactly right.
oxygen is a poison, and people say, "Oh, how can it be a poison?" Now, it's a place to put your electrons in respiration. I understand that, but that's not how it was originally. We learned that, or life learned that, I should say, how to deal with that. So it's so interesting to tell people that, and to say that what was once a waste product that was highly toxic is in fact necessary today. And there's a lot of philosophy to consider with that. But
Yes,
and then furthermore, that brings us to the oxygenation event and banded iron formations. You get different levels of oxygen over time, and you
yes,
precipitate out iron. And so that was happening. But then you talked about snowball Earth multiple times. And what's interesting there is the discussion. We all know about global climate change and higher levels of CO2, but we don't have a geological look at things. And if you look in the geological record, you find out when things got really productive, things were growing very well, and then there was too little CO2 and have the problems in climate. So
exactly.
we're really lucky.
Yes, actually, we are in a very fantastic period of Earth's history, because temperature is more stable, and we have very stable concentrations of oxygen, and that's because, well, this is a matter of debate, of course, but plants, for example, are capable of concentrating tons of carbon in their tissues, and prevents them that prevents that microbial degradation, and that keeps carbon concentrated in tissue or in soils instead of going into the atmosphere. Um, so yeah, like the innovation or the emergence of plants, for example, was very important for us to be here. But of course, before that, there were microbes that were creating photosynthesis, right? Or well, not creating, but uh, generating adaptations to have this fascinating metabolism.
Oh, and one thing that I think it would be good if you take a moment is it's really hard to determine what early microbial life is. And we're used to the idea of looking at fossils. And
Exactly.
and
Yeah.
we had a previous podcast, a former student of mine, Paula Welander, who was able to look at certain biological molecules in the fossil record to get some insights into things. Would you mind telling the people listening and watching why it's hard to study early microbial life at this distance?
Yeah, that's a very fascinating question. And there are multiple reasons. One of them is that this life we are interested in is extremely ancient. As I mentioned, some microbes are like, we know there was microbial life around 3.4 billion years ago, and it's extremely hard to find fossils that are that old. And this is because the surface of Earth is recycling all the time. So there are these subduction areas on Earth where plates are colliding, and one is forced to go under the other one, and rocks just melt. And then they come up to the surface at some point, but that destroys all evidence of microbial life. So we don't have many fossils for microbial life. And um, those that are there, we don't know what they are, because microbes look so similar to each other. So it's very hard to point to a taxonomic identity. So we can say for sure that it's microbial life, or well, not for sure, obviously, nothing in science is for sure, but we can say with some degree of confidence that these fossils are microbial because of the shape, the structures they form, but we don't know exactly what they are. Um, we can say, for example, they are filamentous. They can form filamentous or things like that, but we don't have an identity, or maybe the microbes that form those structures are not alive anymore. Or maybe they are, but it's very hard to say. That's what I'm trying to explain here. And so the microbial fossil record is great. It's great to use it, but I believe we have to use alternative ways to study what happened in the past. And that's why I love using genomic data to study what happened in the past. And this is because DNA, for example, can be considered a biological record because the the uh, the DNA of extant microbes harbor signatures of what happened in the past, and it's very interesting because some people like Desmarais has suggested that the biological record that is in the DNA is highly complementary to the fossil record. So we can link these two records to get a better picture of what happened in the past. And that is where my research falls. I'm very interested in using genomic data to try to understand what happened in the past. And one of the main tools that I use to explore these questions are phylogenetic trees or phylogenomic trees. We coined this term more recently when there was this explosion of genome sequence happening. And phylogenomic trees are diagrams that represent the evolutionary history, especially the diversification events that led to the diversity of organisms we see on Earth today. And well, for those that we have genomes, of course. And they are very interesting because they can tell us, for example, how, for example, the last common ancestor of a certain clade look like. We can figure that out. We can get a hypothesis of how the ancestor of a clade looked like, or what were the adaptations that this last common ancestor acquired. So once we know that, we can, for example, get a timeline of the diversification of microbes that live in a certain environment. So yeah, that's why I really like this idea of using DNA sequences to try to look into the past.
And that's why they're echoes of the past.
Yes, exactly.
And I think it's a good thing to remember. Now, in previous podcasts, we've talked about early life, and we've talked a little bit about what's called the last universal common ancestor, Luca. And the issue with the last universal common ancestor is that is the organism that uses what I call central dogma machinery: replication of DNA, transcription of RNA, translation of proteins. And honestly, I think that's as good as we're ever going to get, because the other one is progenote, and we have no idea what the progenote does, right? We just know it replicates.
Yes.
And maybe that's the RNA world, but that's a different topic. What I like about what you're discussing is you're using what we know today to reflect backward. Just like people would look, for example, at the fossil record to determine that the ancestors of whales looked a lot like cows.
Exactly.
And they changed over large periods of time. So by looking at sequences, you can try to do the same thing. And what I especially appreciate is how you said, and you're right, that people who aren't a big fan of this are ignoring the fact that you do have ways to check with a fossil record and changes in DNA to show that the idea of looking at DNA sequences over time works.
Yes.
If you didn't have organisms for which there was a good fossil record, you couldn't necessarily make that claim. So this is really exciting.
Yes, it's really exciting. This is, of course, these methods that we use are generating hypotheses. So we will never know if this really happened. We can have degrees of certainty, of course. Some hypotheses are better than others, obviously. But I really like this approach because it can allow us to get an idea of what happened in the past. And to some extent, this can give us an idea of what's going to happen in the future with these microorganisms that are so important for the ocean.
Absolutely. And you actually gave me a link to some of your work in this specific area that you said you might want to say a few words about.
Yes, this is a project that I enjoy a lot. This is one of my favorite projects so far. This is a project that I conducted with my PhD advisor, Professor Frank Edward from Virginia Tech, and Joseph Buera, who is also at Virginia Tech. And what we tried to figure out is, can we come up with a timeline of the origin of the most important marine microbes in terms of biogeochemical activities, meaning they are very important for the functioning of the ocean. And second, they are extremely abundant in the ocean. Some more than others, for example, Prochlorococcus and SAR11 are the winners here. But we wanted to get a picture of how these microbes diversified. For example, were they diversifying during a similar period of Earth's history, or each of them have a different evolutionary timeline? That's what we were trying to figure out. And it was very challenging because, as I show in this phylogenetic tree, these microbes are very widely distributed in the tree of life of bacteria and archaea. Some are found in bacteria, Prochlorococcus, SAR11, and others are found in archaea. So, for example, ammonia oxidizing archaea, marine group 1, it's also called marine group 1 or marine group 2. So we needed to come up with a comprehensive tree of life in which we will include all these clades. And after multiple months, and I think a year of running phylogenetic trees and trying to come up with a high quality tree of life, we managed to get it. And it's the one that I will show here. And we did the molecular dating process, which is going from relative distance, which is what we get in a regular phylogenetic tree, to absolute distance in millions of years. So with that, we were able to draw a timeline of when these microbes diversified and the relative distance of one with one another. And what we found is that all these clades diversified during a time span of 2 billion years. Some of them are extremely ancient. As I mentioned before, SAR202 was the most ancient microbe. They've been around for about 2.4 billion years of the ocean's history. And some of them are actually very recent. This is the case of Prochlorococcus. They are more recent in Earth's history. And very interestingly, we found that the physiology that these organisms have right now, or the physiology that we can see in the ocean, is related to some extent with the environmental conditions in which they first diversified. For example, as SAR202, which is this ancient microbe, and marine microbia is another case of a microbe that is very abundant in the ocean. Some of the clades within these groups are very prevalent in oxygen minimum zones. So these are regions of the ocean in which oxygen tends to be extremely low in terms of concentrations. And they are very prevalent there. And this kind of recapitulates the anoxic environmental conditions in which they first diversified. And this goes back to the idea of DNA and even physiology being a biological record. For example, SAR11 diversified during a period in which oxygen was already around, and they need oxygen for their physiology, right? So you can see how biologists harbor signatures of their evolutionary history, right? And that's very fascinating. Another thing that we did is trying to figure out what adaptations microbes acquired during the time of their diversification. And we have found very interesting things. For example, SAR202, which is the very ancient guys, they acquired genes that were related, for example, with the TCA cycle, which is very, very important for carbon metabolism. So that also shows that they are very ancient. So, for example, Prochlorococcus had a very interesting turnover of photosynthesis genes. And this happens when they diversified from their sister clade Synechococcus, which is another picocyanobacteria that lives in the ocean. So when these two diversified, there was a turnover on the photosynthetic machinery, and we were able to see that. Um, so yeah, it was a very, very interesting work.
Do you know what really appeals to me is I'm not going to say that I'm old, but I'm entropically challenged. And what I would say to you is that we were so used to looking at individual things, individual genes. And I'm not saying those aren't important. Those particular approaches do have benefits, but what we can do today, and I can do it with undergrads, where if I do evolution in the lab, that is to say, find things that swim faster or slower, and I'm able to compare their genomes for about $80 to find out specifically what genes, did you hear the plural, what genes have changed. And when you start to think about what took place in ancient times and how with horizontal gene transfer and mixing and matching, you're not looking at one thing anymore. And this is what your career is looking at groups of things. So
Yes.
I would argue there's a term systems biology, and I think this is part of systems biology, but not everybody agrees with me. That's just my opinion. But this is really fascinating, and I think this is great, and I think it leads very well into what projects you anticipate for your grand spanking new lab.
Yes. So the name of my lab is Early Life and Microbial Evolution. And I think that describes very well what we will be doing. So I'm very interested again in early life, what happened in the past that drove the amazing microbial diversity that we see on Earth today. And I have a personal interest for the ocean, of course. So I will continue studying the ocean. And but I'm also interested in more recent evolution. So, for example, um, the exchange of genes in modern environments. So how is that happening? Is there any biological and environmental factor playing a role in the exchange of genes, especially to keep a cohesive species in the ocean? That's a fascinating topic. Uh, but I think my heart is mostly in for the early evolution of microorganisms. And one of the main things my group will be doing is trying to come up with phylogenomic strategies to get better resolution of the past. Because I didn't mention the tricky part of phylogenomics, but there are so many problems associated with these methods. Um, for example, reconstructing a reliable phylogenetic tree is very hard. And the main reason for that is that we don't have a reference tree, right? We don't have a good way to compare, is this tree right or not? So I would like to look for alternative strategies to assess how reliable phylogenetic trees are, because we ultimately depend on these topologies to answer these deep evolution questions. So that's one of the main things my lab will be doing. The second thing is that we need better genomic data. And although we always hear that genomes are increasing on a daily basis, not all of them have a very good quality. That's one thing. And I know it's very tricky. We are doing better. And there are better technologies coming up. Um, but to answer or to explore these deep evolutionary questions, we need good quality data, and that is a challenge. So one of the things my lab will be doing is going to environments that resemble early Earth to try to get genomes that help us look into the past. One of my favorite environments are microbial mats. I have a picture for them. And they're very interesting environments because first, people have suggested that they were very abundant in the past. Um, I'm talking about 3 billion years ago. So it's very likely that we will find ancient life in there. And they have a very high diversity of microorganisms, specifically these ones that are from Guerrero Negro in Mexico. They have a very, very interesting diversity of microorganisms. So we should go for them and try to see what's in there. So generating good quality data, it's one of the main goals of my lab. And the other tricky aspect about genomic data is that we have so many genomes for certain clades, especially those that are human related, nothing against human stuff, but we are interested in the deep past where humans were not still there, right? So we want more genomes from clades that help us explore what happened in the past. And the third part of the research of my lab will have to do with asking and exploring these kind of questions of how did the common ancestor of a certain clade look like? What can we say about the history of life? And these are the main things that my lab will be doing.
So I'm going to show you something now. And what you're looking at is a shell.
Oh,
And
that's nice.
this actually comes from Mexico. I had a
Oh, wow.
student who found what the student described as an anaerobic lake, and this is manganese all over the surface.
Yeah.
And
yeah, these minerals precipitate in anaerobic environments.
right. And so what's important to remember here, which is fascinating to me, is that we tend to overlook the environments that can tell us a lot about our ancestry.
Yes.
How do you study whatever? And a lot of people think of exotic locales. And I'm not saying that where this came from wasn't exotic, although probably it's not the kind of thing that you want to hike next to when you're thirsty, because you're not going to drink from there. But
yes,
the point that I'm trying to get at is that the evidence of the past is all around us at our feet if we take the trouble to look.
Yes, and the ocean is a very interesting environment for those purposes because, as I said, the ocean is extremely ancient. So it has the most interesting microbes to look at.
It's funny because early microbiologists, when they moved away from human-associated microbes, there was a lot of work done by Claude Zobel and other people in the 1920s and 1930s in looking at marine microbiology, and then folks kind of moved away from it. And it always amazed me because there's such interesting things. I, in fact, have my students do, my undergrads, what I call a marine microbiological safari every year to see the interesting things that they find. From things that degrade agar, the agar pitters, to things that are fluorescent under UV light or have funny pigments. It's really amazing. I lots of times have had students find organisms that can only grow in association with others.
Yes, actually, we think that's a very relevant aspect of microbial life.
So,
and we are not aware of that.
No, because we tend to be reductionist, and I understand that. It reminds me, and I think you and I have similar views, so we're laughing at the same thing here. But when people would study biofilms of bacteria, fascinating topic, and they would have one species and they would see how that would happen. And then people like me would say, well, that's not really how biofilms form. They say, okay, I'm going to put three things together. And then I'm rolling my eyes some more. Because on the one hand, that's not how you would learn about a biofilm, but how do you start? That's the question.
Yes, I do agree. And life is very complex, right?
It is. And so, you have to start. You have to start.
Yes, yes. Yes, I do agree.
And I wanted to say one more thing that I thought you would find kind of amusing is that we talk a lot about learning about the past through the echoes that are resounding today. And there's and I'm not going to put this elegantly, so I hope you'll forgive me. There's a trick of architecture where you can whisper in one part of the chamber. You won't hear it 10 feet away, but you'll hear it 100 feet away because of the construction kind of focusing the echoes. Mm-hmm. That's what you need to do.
Yes, exactly. Yeah. Looking into the deep distance.
That's right. You need to actually find that focal point of all those echoes of the past. I think what you're discussing sounds like wonderful opportunities for students on all levels.
Yes.
And I think it's wonderful.
Yes, I would love to hear from people that are interested in this kind of research.
So there you go. I know you'd mentioned that. You also mentioned you might have some ideas about outreach.
Yes, so Santa Barbara is a great place to do outreach activities. There are many schools in the area, and I have always been interested in working with little children uh, because I believe that's the special period of children's life where they can become interested in science. So I want them to be aware that science is there, biology is there, and that's something they can do if they are interested in. So it's better if they show interest early in life, such that parents can focus their interest towards a certain direction. Um, so yeah, that's something I really want to do. I want to go around and talk about microbes with children.
Oh, that's something I'm thinking about. You know, you'll have to invite me down and we can do some Lux art with kids.
Oh, I would love it.
Yeah, and
Thank
and
you.
and they paint with the luminous bacteria, and the next day it glows in the dark and is beautiful to look at. And those are
Thank
marine
you. I would love it.
microbes, and it teaches them that not all microbes are bad, because that's what happens, right? People get that into their heads.
Exactly. That's a very good point. Like changing this view. And one of the main projects I would love to do with students is going around and making them aware of how many microbes are around, because we all, well, even us microbiologists, we go around and we are never aware of how many microbes are under our feet.
Yeah.
And I feel people have to become aware of that, of how much life there is around them, not only in the form of plants or animals, but also microbial, right?
Absolutely. There was a saying about nematode worms. I don't know if you heard this, but this idea was if you made the animals in the Arctic disappear, there would be a three-dimensional image of them in nematodes. And I just move away from that to say, now let's talk about microbes.
Yes,
And I think that's 100% true. There is not a surface on you that doesn't have microbes associated with it.
exactly. I think we all have to be aware of that.
Yeah. So the last thing that I wanted to talk with you about is you said you had a story about how you fell in love with the ocean and microbes in general.
Yes, I love that story. I find it very interesting because I'm from Mexico. I'm from Baja in Mexico. This is a very special place. The ocean down there is beautiful. I haven't seen a more beautiful place than that. Sorry, I'm a little bit biased. Um, but I think I fell in love with the ocean when my parents were taking me for camping trips. So we will do these 5 days trips, and they were so great. And I think that's how I fell in love with the ocean, just by going into nature with my parents. And I think that's something every parent should be doing with their kids. Yeah, if to whenever it's possible, of course. um like being in contact with nature is so important for someone's life, because that made a change in my life, and that pushed me to following a career in science, right? um and so that's the story of how I fell in love with the ocean. So the ocean has always been part of my life since day one, when I was born, because I was born in a coastal area in Mexico. And the reason why I fell in love with microbes is because one day I read about the Great Oxidation Event and how cyanobacteria were the first microbes or the first organisms on Earth producing oxygen. And it's very funny how, after so many years, well, maybe 20 years, I'm here now studying how microbes evolved in the past, right? Um, so yeah, I think cyanobacteria have a very special place in my heart because of that, because they led me to study microbiology eventually. And yeah, so I think having books around and going into nature, it's so important for someone's life. So it's very important to have that in children's lives. That's what I will say.
So there's a book, and I'll put a link to it in the show notes. It's called A Field Guide to Microbiology. Are you? It's by Betsy Dwyer. Have you seen it?
I have not.
I think you would love it because it gives little trips you can take just to see iron oxidizers in different
Oh,
areas.
that's amazing.
Now, what's interesting about all of this is that when you talk about going into nature as a microbiologist, generally speaking, people say, oh, that's not very interesting, but it is.
It is.
If you know what to look at, right?
Exactly.
And how about saying microbes whisper, right? That
Yes.
They whisper, but you have to be tuned to hear it.
Yes, you have to look for them.
You know, it has been just wonderful having you on the podcast, and it sounds like
Thank you so much.
you and I have very similar ideas, and I hope we can interact some more. I actually have some questions for you that I'll send about marine microbiology because I know you'll be interested. Yeah. And I want to wish you and your family and your nascent lab the very best. It's, you know, I do want people to know that when you look at the environment of UC Santa Barbara and you look at the dorms, which are on the beach, I often wonder how anyone gets work done. But you see,
I know.
If you love
we are privileged.
If you love marine microbiology, it's a perfect environment.
Yeah, it's the perfect place to study the ocean. It's a beautiful place. There's always good weather. It's an amazing place. We are all very privileged to be here.
Thank you so much, Carolina.
Thank you so much for having me. This was wonderful.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Carolina Martinez Gutierrez is in the Department of Earth Science at the University of California, Santa Barbara. Beautiful place. Many, many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.